Hello, everyone. My name is Leilani, and today I'll be presenting the blood brain barrier, guardian and gateway of the mind. In this presentation, we'll explore how the blood brain barrier protects the central nervous system, how it maintains homeostasis, and how its selective permeability shapes both health and disease. In the second half, we will examine a scholarly article by Updahe from 2014, which reviews cutting edge drug delivery methods designed to safely cross the barrier. By the end, you'll see how the BBB acts both as a shield and as a future therapeutic pathway. Our objectives today, they're going to be fourfold. First, to review the structure of the central nervous system and understand how it relies on physical and chemical protection. Second, to explain the function of the blood brain barrier and how it regulates what enters the brain's microenvironment. Third, to examine the benefits and limitations of this barrier. And finally, to integrate the scholarly work of Updahe 2014, which evaluates modern drug delivery strategies designed to cross the BBB safely. CNS overview. The central nervous system consists of the brain and the spinal cord. It serves as the command center for the entire body. It integrates sensory input, interprets information, and coordinates motor output. Because neurons have limited regenerative capacity, the CNS requires multiple layers of protection. These include the meninges, which provide physical barriers, the cerebrospinal fluid, which cushions against impact, and most importantly, the blood brain barrier, which regulates the chemical environment that neurons depend on for stable signaling. Protective mechanisms. So, why do we need to protect the CNS? Well, the CNS must be protected because even small fluctuations in blood composition could disrupt the electrical signaling. Toxins, pathogens, and immune cells circulating in the blood could damage delicate neural tissue. The BBB prevents harmful substances from entering the brain and helps maintain very tight ionic and chemical balance. Without this regulation, basic functions such as breathing, movement, and memory would be compromised. Now, let's get into the structure overview, the neurovascular unit. Now, the BBB is formed by the neurovascular unit. It's a coordinated team of specialized cells. The endothelial cells line the capillaries and are sealed tightly by tight junctions, thus preventing unwanted molecules from slipping in between the cells. The pericytes wrap around the capillary walls and provide stability. Next, we have our astrocyte end feet envelope, which is the vessel, and it helps induce and maintain barrier properties. And the basement membrane. This is the anchor for all the structures. And together, these components create one of the most selective and restrictive barriers that we have in the body. Tight junctions and proteins. The tight junction acts like a molecular zipper. Proteins such as claudins and occludins seal the space between the endothelial cells. That's going to be what prevents the paracellular diffusion. These junctions are regulated by intracellular signaling pathways that adjust barrier permeability in response to a physiological need. Their integrity is essential to protect the brain from fluctuations in the blood chemistry. The cellular architecture of the BBB. Excuse me, it's just a BBB. In this longitudinal view, we see how the endothelial cells form the wall. Astrocytes send signals to coordinate barrier functions, and the pericytes help with the vessel repair and regulation. This layered organization allows the BBB to control substances exchange while maintaining communication with neurons. It's an elegant system designed for precision and stability. In the BBB, the selective permeability is a lipid soluble molecule such as oxygen and carbon dioxide. Oh, excuse me, is lipid soluble molecules such as oxygen and carbon dioxide diffuse freely. Essential nutrients like glucose and amino acids require specialized transporters. That's going to be your GLUT1 and your LAT1, respectively. Meanwhile, the efflux pumps, such as the P glycoprotein, remove toxins and drugs from the brain. This combination of diffusion, transporter mediated entry, and active efflux creates a tightly regulated environment, which is necessary for neural function. The protective functions. The BBB limits inflammation by restricting immune cell entry and controls oxidative stress. It maintains stable extracellular ion concentrations, allowing the neurons to fire accurately. So essentially, the BBB preserves and the clarity and consistency of neuronal communication, which means it's protecting the brain from both immediate and long-term harm. Let's talk about some of the advantages. The barrier strength also creates the challenges. On one hand, it protects the brain from toxins, pathogens, and harmful chemicals. And on the other hand, some of the disadvantages is that it blocks roughly 98% of drugs intended to treat neurological diseases, such as Alzheimer's, Parkinson's, and brain tumors. This is why developing methods to safely cross or bypass the BBB has become an urgent priority in medical research. Now, introducing the scholarly study. The scholarly article, Ravikant Upadhyay of 2014, provides a comprehensive review of the drug delivery systems targeting the BBB. It highlights non invasive methods such as nanoparticles, receptor mediated transcytosis, and biochemical carriers. Upadhyay emphasizes that modern approaches focus on working with the BBB's natural transport systems rather than disrupting its structure. Our hypothesis and design. Upadhyay's central hypothesis is that the nanocarriers, which are engineered microscopic delivery vehicles, can be transport drugs across the BBB safely and effectively. The study reviews a wide range of in vitro and in vitro experiments testing these systems. Key strategies include attaching a therapeutic molecule to a receptor, encapsulating drugs with liposomes, and modifying nanoparticles to mimic substances in the brain naturally imports. So, to evaluate these delivery strategies in our experimental models, researchers rely on two major model systems. First, in vitro BBB modules. They use the endothelial cell cultures to stimulate or to simulate barrier properties in a controlled setting. Second, in vivo rodent and primate studies have allowed measurements of actual drug penetration into the brain tissue. These models provide essential data on safety, permeability, and therapeutic potential. The mechanisms of transport. Now, here we can see the summary of major transport mechanisms across the BBB. Receptor mediated transcytosis, where ligands bind to the endothelial receptors and are ferried across the cell. This is going to increase the drug delivery by approximately 30% in preclinical studies. And this is done without damaging the barrier. Chemical pathways involve transferrin and insulin receptors, while transport mediated routes rely on carrier proteins like OATP and ABCG2. These findings demonstrate proof of concept that non invasive drug entry is available. Now, let's talk about a healthy versus diseased BBB. In this figure, it compares a healthy BBB with one that's disrupted by disease. Conditions such as inflammation, stroke, or infection weaken tight junctions and increase permeability. Apede, he notes that the nanoparticles can be engineered to target these compromised regions. Additionally, there are focused ultrasounds when combined with microbubbles that can temporarily open the BBB in a controlled manner, allowing therapeutic molecules to enter without long term damage. Future implications. Future advances will most likely rely on biodegradable nanocarriers, exosomes, and tailor receptors binding to ligands. These methods have the potential to combine BBB protection with precise therapeutic access. Abedes highlight that such innovations may significantly improve treatment for I struggle there, neurodegenerative diseases. And that's going to be your brain tumors and other CNS disorders. The clinical innovation. Let's talk about the brain shuttle. There's one real world example, and that's our Roche brain shuttle technology. This system attaches an antibody module to therapeutic proteins, which allows them to bind natural endothelial receptors and cross the BBB through transcytosis. This represents a major step forward in delivering biological drugs, especially those too large to cross the BBB on their own. And it directly aligns with the mechanism outlined by Upadhyay. Now, in summary, the BBB is highly specialized. It's the guardian of the CNS, maintaining a stable environment for neural functioning. However, its also selectivity also limits the treatment options for the neurological diseases. And as researchers have evolved science, it's beginning to transform this barrier from a strict gatekeeper into a strategic gateway, allowing our therapeutic solutions to reach the brain safely and effectively. Now, I know that was a bunch of information, but just so we can run through it real quick, the blood brain barrier, or the BBB, is like a super protective wall, and that's going to surround your brain. It decides what gets in and what stays out. Good things like oxygen, water, and sugar, they're allowed through because your brain needs them to think and move and stay alive. But the bad things like germs and toxins and most medicines, they're blocked so they can't hurt the brain. Now, your brain is very picky, and it's kind of like a fancy VIP club with a tough security guard at the door. Only the people on the approved list can get inside. So, scientists are trying their best to find smart ways to help important medicines get through this wall when someone's sick. They're creating tiny nanoparticles like special keys that can trick the barrier into letting the medicine inside safely. So the BBB's job is simple: protect the brain, keep it out of danger, let in the good stuff, and finds safe ways to help medicines enter when they need them. It's your brain's personal bodyguard, always working and always watching and always keeping you safe. These are the references that have been used throughout the presentation, including the textbook by Sherwood and the scholarly article by Apede. Thank you for your attention, and you have a great day. If there are any questions or concerns, please feel free to reach out to me. I look forward to hearing from you.